Hello, I'm Jessamine Kuehn, and I'm going to be talking about my research in the White Lab at the Harper Cancer Research Institute at the University of Notre Dame. My project deals with brain cancer, or gliomas, and I work with a specific protein, or a unit in the body that carries out a function. This protein, called isocitrate dehydrogenase 1, or IDH1, is involved in metabolism, or converting the energy from food that you eat into chemical energy that can be used by the body. So proteins, like people, carry out their jobs in different ways. Some proteins might work individually, like how some people might work individually at a desk job, while others might work in pairs, like how it might take two people to lift a heavy object. But if one of those two people is sick or weak, then they can't lift that heavy object or carry out their original function. Similarly with proteins, if one is changed or has a mutation, they might not be able to do their original function. Now in the case of the protein that I'm working with, the protein that has a mutation isn't just unable to do the original job, but it's now able to carry out a new function. So it's not just a weak protein, but it's a bad protein. It's a traitor protein now doing a different job for a different agency, in this case, participating in a new reaction that produces a harmful chemical that helps cancer rather than helping the body. So my question is, how does this harmful protein do its harmful job? Previous work has shown that more of this harmful chemical is produced when both the bad protein and the good protein are present, rather than when only the bad protein is present. So that leads to the hypothesis that it takes a good protein and a bad protein working together in a double protein structure, or a heterodimer, to produce this harmful chemical. Other previous work has shown that more of the harmful chemical is produced in more acidic environments, leading to this second hypothesis that more of this double protein structure will be formed in more acidic environments. So to test these two hypotheses, we ran experiments that looked for the protein's size. And we knew that if we saw a size that's double the known size of the protein, then we have some of that double protein structure going on. And we did find that there were more of that doubly sized protein, or the double protein structure, in more acidic environments. So that supports our hypothesis that it takes a good protein and a bad protein working together, forming a heterodimer in a more acidic environment for the bad protein to do its job. So these results further our understanding of this mutation that's commonly found in brain cancers. It can help us to have earlier diagnosis of brain cancer if, for example, we can detect the presence of this double protein structure, or it can lead to treatment of brain cancer if we can target the double protein structure and prevent it from producing the harmful chemical. So this project took about 2 years, and like any research project, it had its ups and downs and problems that arose that needed troubleshooting. One of the most confusing results was when we found that a negative control that didn't contain the protein had high levels of activity, and after some troubleshooting we found that the reason for that was one of the substrates for the reaction, alpha-ketoglutarate, was more concentrated than we thought it was, so we remade the substrate stocks and that solved the problem. But it was very exciting when we got our results that supported our hypotheses. So research can be challenging, but it is feasible, and it is rewarding when it does give results. So it is possible for new researchers to conduct meaningful research. It takes time and troubleshooting, but ultimately brings us closer to improving human health. Thank you.